This is a video to recap the stages of gene expression and to look at the production of proteins in cells. Okay, so when we're talking about gene expression, we are talking about genes within our DNA becoming switched on, and this just means that we start making these proteins that those particular genes code for. So we look at this little section of DNA here. This is our gene. What we are doing in gene expression is we are making the protein that that particular gene codes for. And we do this through three main stages. So our first stage that we can see here is transcription, and this occurs in the nucleus. We will look at this in a lot more detail in a second, but just thinking about the names at the moment. And we can see that we're making a single-stranded copy of DNA. We'll remember that from Nat5, we're making mRNA. The second stage also occurs in the nucleus. We call this RNA splicing. And this is where we remove any non-coding regions, anything that is not going to be part of our final protein. And then the last stage we call translation, and this occurs at the ribosome. This is where we make our polypeptide. Okay, so next thing that I want to look at just before we move on is we're going to look at the differences between DNA and RNA. If we look at our DNA here, we can see that we've got our base, our sugar and our phosphate. Our sugar here is deoxyribose sugar, and our bases are A, T, C and G, and we know that DNA is double-stranded. Whereas when we look at RNA, it is made of ribose sugar this time. And instead of a T, we're going to replace that with U or uracil. And also RNA is single-stranded. So now we'll start to look at those different stages in a bit more detail. Okay, so starting to look at the process of transcription, and this stage occurs in the nucleus, and it involves the synthesis of mRNA from a section of DNA or our gene. And this stage involves the use of an enzyme, the only enzyme that we're going to talk about in gene expression, and it's called RNA polymerase. And it's really important that we don't confuse the function of it with DNA polymerase and DNA replication. Now, what RNA polymerase does is it's going to move along our DNA and it's going to unwind the double helix, or unwind our little section of DNA. And it is going to break the hydrogen bonds between the bases, and it's going to add these RNA nucleotides to the 3' end and synthesize our primary mRNA transcript by complementary base pairing. So we know that T goes with A. A goes with T in DNA, but in RNA we change that to uracil. G, G. And it's just going to move along, adding those complementary bases. And hydrogen bonds are going to form between those complementary base pairs. And those are going to allow the bonds to break and our primary mRNA transcript to break away from that DNA. So we've now synthesized our primary mRNA transcript, but before it can leave the nucleus to take that copy of DNA to the ribosome, we need to ensure that our transcript only contains information that's required to make our protein. And we do this by a process called RNA splicing. So if we look at DNA, it actually contains lots of non-coding regions, and those can be involved in things like regulating transcription, but they actually don't contain any information to make our protein. And we call these introns. So introns are non-coding regions. And we've got other regions that are going to code for protein, and we call those exons. And I always think introns stay in the nucleus, exons are going to exit the nucleus. So let's say if we look at this AUG at the start of our primary mRNA transcript, let's say that is an exon, that is a coding region. And GCG, that's non-coding, that's our intron. UGU, let's make that an exon, and UAA, that is going to be an exon as well. Now what we do is we're going to remove these introns by RNA splicing, so we're going to cut them out, and what we are going to be left with is all of these exons. And you can see that this new transcript is a lot shorter, it contains exons only, and we're going to call this our mature mRNA transcript. Now, what you could be asked is, why is our primary mRNA transcript shorter than DNA? It is because we're only transcribing one gene, so we've only unwound a little section of our DNA and we've just transcribed that one single gene. And what else you could be asked is, you could be asked, why is the mature mRNA transcript shorter than our primary mRNA transcript? And that is because it contains exons only. Okay, so last stage is translation, and this occurs at the ribosome. This is where we're going to synthesize our polypeptide chain. And we can see that our mature transcript containing just those codon regions has attached to our ribosome. And it consists of these triplets of bases. We call these codons, and these are going to code for specific amino acids. Now, the other little structure that I wanted to show you just now is this here. So we've got another type of RNA, we call this tRNA or transfer RNA, and it consists of an anticodon that we can see here, and we've got this amino acid attachment site. So to start forming our protein, this tRNA is going to collect a specific amino acid and it is going to transport it to the ribosome. So this anticodon on the tRNA is going to complementary base pair to our codon on the mRNA, and in doing so we're going to bring that specific amino acid to the ribosome. And we have these weak hydrogen bonds forming in between the codon and the anticodon. Same thing's going to happen for the other ones. So again, bringing that specific amino acid to the ribosome. And last one, bringing our last amino acid to the ribosome. Now, to form our polypeptide, we're going to form these strong peptide bonds in between our amino acids. And once we've done that, our polypeptide can just detach and it can go and carry out its function in the body. The last thing that we need to know is that these polypeptides aren't just going to stay linear like I've drawn here. They are going to fold to form this 3D shape. So it'll start to look a little bit more like this. And this just occurs through weak hydrogen bonds, and we've got these other interactions that can occur in between our amino acids. So alternative RNA splicing is another process that can occur in the nucleus, and this is the process by which we make many proteins from one gene. If we look at our gene here, our DNA, we can see that we've got multiple codon regions or exons. So we've got exon 1, 2, 3 and 4. And once we have made our primary mRNA transcript, you can see that 2 different proteins have been made from that one transcript. We've got protein A and we've got protein B. And this is completely dependent on what exons we retained. So for protein A, we've got 1, 2 and 3. We didn't retain exon 4. Whereas in protein B, we've got 1, 2 and 4. We didn't retain exon 3. So this is really advantageous for our cell, as it means we can make many different proteins from one single gene, depending on what exons we retain. So there are 3 types of RNA that we need to know about. mRNA we've already talked about, that is your single-stranded molecule, and that's going to carry a complementary copy of DNA from the nucleus to the ribosome. tRNA we talked about being used in translation, so remember that had an anticodon and it had an amino acid attachment site. And remember, those anticodons were going to complementary base pair to a codon on your mRNA. And then the last one is rRNA or ribosomal RNA. And that, all that we need to know about that is that rRNA and protein is going to be what makes up our ribosome. So there's a few skill questions that can come up for this topic, and a lot of the time they revolve around these mRNA codon tables, and they just show the positions of the bases on the codon and what the corresponding amino acid would be. So we could be asked something like, which amino acid would be coded for by DNA sequence GAC? So this is an mRNA codon table, so we need to work out what the mRNA sequence would be first. So G is going to go with C, A is going to go with U, and C is going to go with G. So if we look at this bit here, we're looking for our first base of codon, and that is a C. So we know we're in the right-hand side of the table. Second base of codon is going to be U, so just circle that. And then the third base of the codon is going to be G. So we end up in this little section here, and our amino acid is going to be leucine. So same skill, but we're looking at a slightly different table this time. We're being asked which amino acid would be coded for by DNA sequence ATC. So because we're looking at an mRNA codon table, again working out the mRNA first. So we're UAG this time. First position, we have U, so we know we're in these first set of rows. And second position, we are A, so we know that we're looking in this little section here. And then third position, we have G, so we're looking at this stop codon. And that's it for gene expression. Hopefully that has been helpful.